In this talk, we're going to learn about different types of immune cells. So we're going to focus on the origins of immune cells. We're going to look at different types of immune cells, which include cells of the myeloid and the lymphoid origin. All blood cells, including immune cells, arise from the hematopoietic stem cells that are found in the bone marrow. The hematopoietic stem cells are pluripotent because they can form a wide variety of cells that are listed here. Let us now look at how these different types of cells arise from the hematopoietic stem cells. The pluripotent hematopoietic stem cells undergo differentiation to form all the cells found in blood, which include the immune cells. So one pathway is that the pluripotent hematopoietic stem cell can undergo differentiation to form a common myeloid progenitor. This common myeloid progenitor cell can undergo further differentiation to form different cells of the myeloid origin. These include the red blood cells and platelets, as well as neutrophils, eosinophils, basophils, monocytes, mast cells, macrophages, and dendritic cells. On the other hand, the pluripotent hematopoietic stem cell can differentiate to form the common lymphoid progenitor. And these common lymphoid progenitors can undergo differentiation to form dendritic cells, innate lymphoid cells, or B or T lymphocytes. Now, when we look at these two different paths, we can see that dendritic cells are common, and hence dendritic cells can arise from common myeloid progenitors or common lymphoid progenitors. Now, when we look at which cells are involved in innate immune responses, then all the cells that are in that orange box are the ones that are involved in the innate immune response. On the other hand, when we look at cells that are involved in the adaptive immune responses, then all the cells that are in that purple box play a role in the adaptive immune response. Now, what should be noted is that dendritic cells actually play important roles in both innate and adaptive immune responses because they act as a bridge between the innate and the adaptive immune response. So let's look at some of these cells in more depth. We're going to start off looking at myeloid cells, and we're going to start with the erythrocytes and platelets. Now, myeloid cells arise from the common myeloid progenitor. And so erythrocytes and platelets will arise from the megakaryocyte erythrocyte progenitor that itself arises from the common myeloid progenitor. Now, erythrocytes are red blood cells, and they arise from erythroblasts. So if you try to map the different stages of differentiation, it starts with the hematopoietic stem cell that forms a common myeloid progenitor, which then forms the megakaryocyte erythrocyte progenitor, which then forms the erythroblast, and finally the erythrocytes. Erythrocytes, or red blood cells, are the most numerous cells that are found in blood. So we have about 3.5 to 5.5 billion erythrocytes per milliliter of blood. These cells do not have any nuclei, and their primary function is to carry oxygen to different parts of the body and collect carbon dioxide that it will then take back to the lungs. They do not play an important role in immune responses. Platelets, on the other hand, arise from megakaryocytes. So they're also myeloid cells, and there are numerous platelets that are also present in blood. Now, platelets are not considered as intact cells, but they're considered as cell fragments and play very important roles in forming clots. Clot formation is really important because it prevents bleeding, and so you won't have blood loss when there is a cut that results in damage to blood vessels. We're now going to look at leukocytes, or white blood cells, that are of myeloid origin. So let's first look at the granulocytes. Now, the common myeloid progenitor can undergo differentiation to form the granulocyte macrophage progenitor. This granulocyte macrophage progenitor can undergo further differentiation to give rise to a wide variety of cells that include the granulocytes. There are three types of granulocytes, which are the neutrophils, eosinophils, and basophils. They're also called as polymorphonuclear cells or PMN cells. They're called granulocytes because when we look at the picture, especially the one to the left, we can see these tiny dots in the cytoplasm, which are basically granules. Let us first look at neutrophils. Neutrophils have these distinct nuclei that allow us in their recognition. There are about 3 to 6 million neutrophils per milliliter of blood, and when we stain the blood cells, then this is what a neutrophil looks like. And when we look at the nucleus, which is shown in this purple staining or dark pink staining, it has these multiple lobes. And hence neutrophils are called as multi-lobed nuclear cells. They play very important roles in innate immune responses by recognizing, ingesting, and killing pathogens. They're able to ingest or uptake pathogens by phagocytosis. So neutrophils play a very important role in the clearance of pathogens. Eosinophils are granulocytes, so they're similar to neutrophils, but they do have some differences. They're similar because they have granules in their cytoplasm, the way neutrophils have. But when we look at their nuclei, the shape of the nucleus is different. So when we look at the picture, we can see that the nucleus has two lobes that are indicated by these arrows. Now, eosinophils can also ingest pathogens by phagocytosis, but they can't do that as effectively as neutrophils are able to do so. They do undergo a process called degranulation, where they release these granules that are present in their cytoplasm. These granules have toxic chemicals in them, and so by releasing them into the environment or in the vicinity of a pathogen, they're able to kill the pathogen. Eosinophils are known to play a very important role in killing helminths or parasitic worms. They are also known to play roles in allergies. Basophils are granulocytes that are present in far fewer numbers than the eosinophils and the neutrophils. Their nuclei are shaped differently, and so they have bean-shaped nuclei. They can also ingest pathogens by phagocytosis, but once again, they're not as effective as neutrophils. Basophils have granules in their cytoplasm and can undergo the process of degranulation similar to eosinophils. When this happens, they are able to release molecules like histamine and heparin that are present in the granules. Basophils are known to aid in fighting parasites like ticks and worms, but they also play roles in allergies and inflammation. Mast cells are cells that arise from the common myeloid progenitor that undergoes differentiation to give rise to the granulocyte macrophage progenitor that eventually will give rise to a mast cell. Now, mast cells, even though they are made in the bone marrow, they don't mature there. They will leave the bone marrow as immature precursors, and after migrating to peripheral tissues like the skin and the airways or intestines, that is where they undergo maturation. Mast cells are known to respond directly to pathogens, but they will also produce signals that modulate immune responses. Mast cells are also known to release granules that contain histamine and proteases. So similar to the eosinophils, the basophils, and the neutrophils, they also have granules in their cytoplasm. Mast cells are known to play vital roles in allergies and inflammation as well. Monocytes are leukocytes that also arise from the granulocyte macrophage progenitor. There's about 100,000 to 600,000 monocytes per milliliter of blood. So there's a huge variation in the numbers depending from person to person. Monocytes can be identified by a characteristic kidney bean-shaped nucleus. So this is a little similar to basophils that also have a bean-shaped nucleus, but the main difference is that monocytes do not have granules in their cytoplasm. There are two main types of monocytes, the classical and the non-classical. The classical monocytes differentiate into macrophages after they enter tissues. So monocytes are normally found in blood, But during an infection process, they can leave the blood and enter into tissues where they're going to differentiate to form macrophages. This is what classical monocytes do. Non-classical monocytes are monocytes that will patrol the endothelium, and that's why they're also called as patrolling monocytes. In case you didn't know about this, but endothelium basically means the walls of the blood vessels. Monocytes can recognize and ingest pathogens by phagocytosis, eventually killing them. Thus, they play similar roles to neutrophils and hence are able to clear pathogens. Macrophages are another type of myeloid cell that arise from the granulocyte macrophage progenitor cell. Macrophages are phagocytic cells that are usually present in tissues, so we don't find macrophages in blood. Macrophages can arise in two ways. The first way is when progenitor cells enter tissues during embryonic development. Another way by which macrophages can be formed is from circulating monocytes. So when circulating monocytes enter tissues due to signals of an infection, they can differentiate in the tissues to form activated inflammatory monocytes or macrophages. So this way we have two ways of forming macrophages. Now macrophages are able to kill pathogens by ingesting them through phagocytosis. Not only do they kill pathogens, but macrophages can also kill infected host cells, also through the process of phagocytosis. Macrophages can also induce inflammation by making molecules that promote the process of inflammation. Macrophages can also have distinct names based on the tissue that they reside in. For example, Kupffer cells are the names of macrophages that are found in the liver, while microglial cells are macrophages that are found in the brain. Alveolar macrophages, on the other hand, are macrophages that are found in the lung. So this way we can have tissue-specific names for macrophages. Dendritic cells, which are shown in that purple box, are phagocytic cells that are present in most normal tissues. What is unique about dendritic cells is that they can arise from either the common lymphoid progenitor or the common myeloid progenitor. Usually most immune cells will arise from either one of them. Both progenitors can give rise to the same type of cell, but this is not the case of dendritic cells. So either the common lymphoid progenitor or the common myeloid progenitor can undergo differentiation to give rise to an immature dendritic cell that will undergo further differentiation to form the mature dendritic cell. Now, dendritic cells are able to ingest and destroy pathogens by phagocytosis, but their main job is to sample the environment, and they tend to do that by micropinocytosis. As they sample the environment and are able to take material through macropinocytosis, or they are able to uptake pathogens by phagocytosis, they display components of the pathogens or antigens that are present on the pathogens on their cell surface. And these antigens are then recognized by cells of the adaptive immune response. Thus, dendritic cells play an important role in both innate and adaptive immune responses. And that is why it is thought that dendritic cells act as a bridge between innate and adaptive immune responses. Now, unlike all the other cells that we saw before, innate lymphoid cells arise from the common lymphoid progenitor. They reside in peripheral tissues and are able to mediate inflammatory responses by producing molecules in response to pathogens. There are different types of innate lymphoid cells, but the group that is most well studied are the natural killer cells. The natural killer cells are large lymphocyte-like cells, and they are able to recognize and kill cells that are infected with either viruses or these cells are tumor cells. Thus, the innate lymphoid cells, they play important roles in early innate responses to viral infections. When we look at the picture, the way we can observe an innate lymphoid cell is that it has a pretty large nucleus compared to the cytoplasm, and it also shows the presence of granules in that cytoplasm. Let us now look at the lymphocytes, which play an important role in the adaptive immune responses. So there are two types of lymphocytes, and the first one we're going to look at are the T-lymphocytes. T-lymphocytes are produced in the bone marrow, but they leave as immature precursors and migrate to the thymus. In the thymus, they will complete their development. There are many different subsets of T cells that are found, and we're going to be looking at three main categories, which are the helper T cells, the cytotoxic T cells, and the regulatory T cells. Now, T cells normally are not in an active state. They are what is called as a naive state, and so the cells have to be activated to be functional. The activation of a T lymphocyte occurs when a T cell receptor that is present on the T lymphocyte is going to bind to an antigen. T lymphocytes are known to play important roles in humoral as well as cell-mediated immunity. The next type of lymphocyte are the B lymphocytes. B lymphocytes are produced and they mature in the bone marrow. They synthesize a cell surface receptor called as a B cell receptor. The B cell receptor is also called as an immunoglobulin or an antibody, and it is able to bind to specific components of a pathogen, which are basically the antigens. Once the B cell receptors bind to the antigen, the B lymphocytes get activated. So similar to the T lymphocytes, they are usually present in a naive state or an inactive state. Once the B cells are activated, they can secrete antibodies. So this is when the B cell receptor is cleaved off the cell surface and it is now free to move in the environment. B cells, since they make the antibodies, are the ones that play a very important role in humoral immune responses. With this, we come to the end of the origins of different immune cells and the cells that are formed from the common myeloid progenitor and the common lymphoid progenitor.